Good day, everyone. Welcome to another lecture in the series Medicinal Chemistry 1. And today we're going to talk about adrenergic neurotransmission. So what are these adrenergic drugs? Well, they exert their principal pharmacological and therapeutic effects by either enhancing or reducing the activity of the various components of the sympathetic division of the autonomic nervous system. So the substances that produce effects similar to stimulation of sympathetic nervous activity are known as sympathomimetics or adrenergic stimulants or adrenergic agents, and those that decrease sympathetic activity are referred to as sympatholytics or anti-adrenergics or adrenergic blocking agents. So just to say that these drugs, whether or not they produce their effects similar to or they decrease the sympathetic activity. So that's the distinguishing factor there. So this should be a familiar image from last class, right, where we looked at the autonomic versus the somatic nervous system. So here we are looking at the adrenergic receptors, right? This is what we're looking at today. And we're looking at these neurotransmitters, so or epinephrine and or norepinephrine, etc. So these adrenergic agents, they act on adrenergic receptors, as we just saw, or affect the life cycle of these adrenergic neurotransmitters, so including, as I just mentioned, or norepinephrine, otherwise called noradrenaline, right? So throughout the lecture, please note that when you see NE, I refer to norepinephrine or noradrenaline. When you see E, I'm referring to epinephrine or adrenaline. And when you see DA, I'm referring to dopamine. So these adrenergic agents, they act on adrenergic receptors or affect the life cycle of these adrenergic neurotransmitters. All right, so we're looking now at the adrenergic neurotransmitters, their structure and physiochemical properties. So our norepinephrine, epinephrine, and dopamine are chemically catecholamines. So we must know the structure of these catecholamines, right? So this is our catechol unit, and these are, well, the catechol portion refers to all organic compounds that contain orthodihydroxybenzene, right? So this is a benzene, and the hydroxyl groups are arranged ortho to each other, and so this name should be clear. So catechol nucleus is equal to orthodihydroxybenzene. And catecholamines, they have the catechol nucleus as well as the ethylamine group. So this portion here, so we have 2 carbons that joins the amine, the amino portion, to the catechol. Well, this is just a benzene ring, but if you have the catechol portion, like dopamine here, we have the catechol and we have the ethylamine. So collectively we call these catecholamines. And if we look at these structures here, we see the catechol portion and we see the ethylamine portion, albeit they are functionalized, but the basic structure of being a catecholamine should be obvious. So this leads us to the first question, which says, draw the structure of isoproterenol and identify the catechol and the ethylamine moieties. So just pause and go back, draw the structure, and then just circle the catechol portion and the ethylamine portion. So if you drew the structure and if you circle the catechol part, which is you're seeing it here in, covered in orange, and we have the ethylamine portion covered in green. So if you got that, congratulations. So my follow-up question 1 is, distinguish between sympathomimetics and sympatholytics. All right, so ensure that you answer these questions and come prepared to answer it in class. All right, so now we're going to talk a little more about epinephrine. So we have the structure here, right? And looking at the structure, you're seeing some polar groups in the form of, the hydroxyl groups. So it is polar, and because it is polar, it is water soluble, and it is a weak base with a pKa of 9.9 because of its aliphatic amino group. So we have the secondary amine portion here, which is responsible for it being a weak base. And it is also a weak acid with a pKa of 8.7 because of its phenolic hydroxyl group. So we have 2 phenolic hydroxyl group, which makes it weakly acidic. So it can be predicted that ionized species, that is the cation form of epinephrine at physiological pH, is predominant. So this largely accounts for the high water solubility of this compound as well as other catechol amines. So in other words, if all of the catechol amines have similar characteristics, and because it is an ionized species at the physiological pH, then it is highly water soluble. We can therefore predict that epinephrine has poor absorption and poor central nervous system penetration, right? So because to have CNS penetration, it has to be more lipophilic. So let's look at the stereochemistry of epinephrine and norepinephrine. So they both possess a chiral carbon, right? So if we remember from our chemistry, right here, this center here for both of them, so that carbon that has 4 different things attached to it is called our chiral carbon. And because we have a chiral carbon, they each can exist as an enantiomeric pair of isomers, right? Meaning we can have R or S. So the enantiomer with the R configuration is biosynthesized by the body and possesses the biological activity. In other words, there is the potential to have the R and the S configuration. However, the body produces, in other words, it is biosynthesized by the body and possesses the biology. The R version is biosynthesized and possesses the biological activity exclusively. Now, this R configuration of many other adrenergic agents also contributes to their high affinity to the corresponding adrenoceptors, right? So the important point here is that we have a chiral center, and we're saying that the R is the configuration that is biosynthesized by the body and therefore possesses the biological activity. So let's do a little short review of the RS, right? So if we recall that we're looking at this chiral center here, right? So we're looking at the atoms that are attached to that chiral center. So we see the oxygen here, and we see the hydrogen. Good. We see the oxygen, we see the hydrogen, we see a carbon, and we see another carbon. So we have four atoms that are attached to it. So we're looking to prioritize the attached atoms based on atomic number, right? So here we can see that clearly the oxygen is the highest priority and the hydrogen would be the lowest priority. But for the two carbons here, they are both carbons, so we have to look at what those carbons are attached to, right? In the case of this carbon, it is attached to a nitrogen, right? The other is attached to carbons, and this carbon is attached to a nitrogen. Therefore, it has the higher priority of the two carbons, right? So again, if we rotate, so we ensure that the group that has the lowest priority, in this case the hydrogen, is towards the back, right? Which it is towards the back, and therefore we trace from 1 to 3. So 1, 2, and 3, right? So because it is clockwise, then the configuration is not S but R, right? So if it was anti-clockwise, it would be S, and because it is clockwise, it is R, right? So this is the isomer that is biologically active. All right, so in terms of how these isomers interact with the receptors, right? So we're looking at the 3-point model of drug receptor interaction. So looking at the chiral center, right? We're looking at the hydroxyl group here, the hydrogen, the amino portion, and our benzene ring, and we're seeing the interactions here with the receptor, right? So this is a representation of our adrenaline binding to a receptor. So we have the interaction here between the oxygen and the receptor, and the nitrogen, the amino portion, and the receptor, and the hydrophobic interaction here, right? The S, this slide is trying to say that the S enantiomer is not active because of the lower binding energy, right? So clearly, because the hydroxyl group is oriented differently, it's not able to have the interaction that we see here. And that is why the R isomer is the one that has the biological activity. It's a very interesting study here. It's not matched. It matches here. So here we have an oversimplified version of the different groups, and we see the matching. So each group, the hydroxyl group matches, the hydrogen, all of the group matches with the R enantiomer, while with the S, it's just not matching up with the receptor, which is why it is not biologically active. So that leads us now to question 2, which says, which isomer, R or S, of epinephrine or norepinephrine is active? And part B says, draw the S isomer of norepinephrine. All right, so the answer is you guessed it, the R isomer possesses the biological activity, and part B, we're asked to draw the S isomer. So I have it there. I have drawn the S isomer. So the only difference is that this is a hash as opposed to the R being a wedge, right? So how we do it, or one way to do it, is to assign the priorities, again 1, 2, 3, and then the hydrogen would be coming towards us, right? So every time we see the wedge, that group is coming towards us. The hash is behind the plane. So when we do the priorities and when we connect 1, 2, 3, it's a R. But because the lowest priority group is coming towards us, then it's the opposite of that, right? So this is now S. So that is one way to do it. The other way to do it is to do the rotation in your mind and then assign the priorities, and then follow the numbers, and if it's clockwise then it's R, if it's anti-clockwise then it's S, right? So this is an easy way to, if the group that should be at the back is at the front, is coming towards you, then you go ahead and assign the priorities and then follow the movement from 1, 2, 3, and whatever you get, the actual configuration is the opposite of that. All right, so that is one way to do it. So our follow-up question 2 says, draw the ionized form of epinephrine and explain why it, quote, has poor absorption and poor CNS penetration. Okay, so think about it and try to answer that. All right, so we're going to another portion of the presentation, right? And just to give an overview, because so far we have looked at the autonomic and the somatic nervous system and the parasympathetic and the sympathetic division, but what are they connected to, right? So firstly, we have our CNS, right, which is responsible for integrating, processing, and coordinating sensory data and motor commands. And that is connected to our peripheral nervous system, right, which is responsible for maintaining communication between the CNS and the rest of the body. Okay, and then that therefore is connected to our motor or efferent division, which conducts signals from the CNS to the peripheral tissues, and they are connected to our autonomic and somatic, etc. Right? So we have, it's like a circle. We have, sensors that get the information, then it goes to the CNS, and in CNS there's also movement to and from the CNS, right? So it's just good to have an appreciation of the whole, of the entire system and not just a portion. So just going back, as we say, repetition is key. So just going back to some definition and some key phrase and words that we have been looking at. So the sympathetic and the parasympathetic divisions typically function in opposition to each other. But this opposition is better termed complementary in nature. So they're not fighting each other. They're complementary in nature rather than antagonistic, right? So for an analogy, one may think of the sympathetic division as the accelerator and the parasympathetic division as the brake, right? So one is pushing us to go forward, the other one is saying, hold up. The sympathetic division typically functions in actions requiring quick responses, right? While the parasympathetic division functions with actions that do not require immediate action, which we know already. So in other words, the sympathetic division is basically responsible for fight or flight, and the parasympathetic division responsible for rest and digest. And we know this already based on, last class and even the previous slides, right? This is a nicer image which basically says the same thing. So again, it starts from the autonomic nervous system and the somatic nervous system. We see our divisions here and we see our ganglion, right? And in this case, we have our adrenal medulla and we have our neurotransmitter in the form of acetylcholine. And then eventually we have our epinephrine and norepinephrine in our blood vessel, and then that all of that is connected to our smooth muscle or glands and our cardiac muscle. So we already have an idea of all of this, based on last class and previously, but as I said, repetition is king. And so we need to be repeating so that we can get it cemented. All right, and just in case you have no idea where the adrenal gland is or where it is, just above the kidney, right? So when we refer to the adrenal medulla here, then that is where it is. All right, so just in case you might be wondering, what is fight or flight? Well, the fight or flight response, also called the acute stress response, was first described by Walter Cannon in 1929, way back when. So his theory states that animals react to threats with a general discharge of the sympathetic nervous system, priming the animal for fighting or fleeing, right? So when we look at this image here, the fight is saying stand your ground, defend your position, attack, dig in, persevere. While the flight is saying give way, retreat, discard, remove yourself, give up, move on. And I'm sure we can think of some Jamaican ways to express this, right? So this response was later recognized as the first stage of a general adaptation syndrome that regulates stress responses among vertebrates and other organisms. All right, so what actually happens during these fight or flight responses? Well, these catecholamines that we just looked at earlier, these hormones, they facilitate immediate physical reactions associated with a preparation for violent muscular action. So what happens? You have acceleration of heart and lung action. You have inhibition of stomach and intestinal action. You have constriction of blood vessels in many parts of the body. You have liberation of nutrients for muscular action. You have dilation of blood vessels and muscles, inhibition of tear glands and salivation. You have dilation of your pupil, relaxation of your bladder, inhibition of erection. So all of these things happen under the fight or flight response. Okay, so for noradrenaline, you have glycogen in the liver is converted to glucose. You have increased pupil size, sweat glands produce more sweat, heart rate increases, adrenal medulla releases adrenaline. You have bronchial tubes in the lungs dilate for greater oxygen intake. And for adrenaline, you have boost of oxygen to brain and muscles, and you have suppression of digestion. All right, so now we're going to go a little bit more into the biochemistry of these adrenergic receptors. Right, so in 1948, the adrenergic receptors were subdivided into alpha and beta by alkist. So the distinction was based on sensitivities of different organs to catecholamines of closely related structure. And so the regulation of the functions of the different organs depends on, to a greater or lesser extent, on alpha or beta receptors. So we're gonna go into it's a little bit more, so we have our adrenaline and our noradrenaline, we have our two alpha receptors, we have a few beta receptors, and at the end of the day you have smooth muscle contraction and you have smooth muscle relaxation, etc. All right, so the alpha receptors are located postsynaptically, so we need to understand what this means. And if we don't remember, just go back to the previous lecture, at sympathetic neuroeffector junctions of many organs. So in general, alpha receptors, they mediate excitation or increased activity of the effector cells. So vascular smooth muscle is an important site of alpha receptors, and the SNS or sympathetic nervous system activity maintains vascular tone and thus blood pressure by maintaining a tone of neurotransmitter on vascular alpha receptors. So beta receptors, on the other hand, are located postsynaptically at sympathetic neuroeffector junctions of many organs, and in general, they mediate relaxation or decreased activity of the effector cells. So let's go back to the alpha. So the alpha, in general, mediate excitation or increased activity, while the beta mediate relaxation or decreased activity, note in general, of effector cells. So thus blood vessels dilate and uterine smooth muscle relaxes in response to activation of beta receptors. So the heart muscle is an important exception to this rule. Activation of beta adrenal receptors in the heart increases the automaticity and contractility of all parts of the heart, which is why over here we have alpha, smooth muscle contraction, but when we reach beta, smooth muscle relaxation. However, for the heart muscle, we have contraction, right? So we must note that important exception to the rule. So what are the types of adrenergic receptors? So alpha adrenergic receptors are adrenergic receptors that respond to norepinephrine and to such blocking agents as phenoxybenzamine. So they are subdivided in two types. So we have our alpha 1, which is found in smooth muscle, is found in the heart and the liver, with effects including vasoconstriction, intestinal relaxation, uterine contraction, and pupillary dilation, as opposed to alpha 2, which is found in the platelets and vascular smooth muscle, nerve termini, etc., right? And inhibition of norepinephrine release and of insulin secretion. All right, so we have alpha 1 and we have alpha 2 located in these different areas. For the beta, it responds particularly to epinephrine and to such blocking agents such as propranolol. So there are 3 types of beta, and they're designated beta 1, beta 2, and beta 3. And the beta 1 adrenergic receptors are located mainly in the heart, right? And beta 2 are located mainly in the lungs, GI tract, liver, uterus, etc. And beta 3 receptors are located in fat cells. So what do the receptors do, right? So we mentioned this already, so this is we're kind of summarizing this. So activation of the alpha receptors leads to smooth muscle contraction. Activation of beta 2 receptor leads to smooth muscle relaxation. Activation of beta 1 receptor leads to smooth muscle contraction. So that is the exception there, right? And then we have this table which kind of further breaks it down, the different organs and the predominant adrenal receptors. Notice that they are located elsewhere, but we're looking at the predominant adrenal receptors, the effect of their activation, and the physiological effect, right? So at this point, I know you guys are going to ask me, do I need to know this table? Well, I mean, what you need to know the effect of activation, definitely, right? For all of them, right? And you need to know the heart, definitely, and just a general idea of, you know, the alpha. You need to be able to know where the alpha 1 receptors are, the mainly, right? Where the alpha 1, alpha 2, and all the betas, right? Where they're located. And this is easy to remember, the effect of the activation. All right, so this leads us now to question 3, which says, fill in the blanks. Activation of alpha 1 receptors leads to. Activation of alpha 2 receptor leads to. Activation of beta 1 and 2 receptors leads to. All right, so pause, answer. All right, so the activation of both alphas leads to smooth muscle contraction. Activation of beta 1 leads to smooth muscle contraction, right, especially in the heart. And activation of beta 2 receptors leads to smooth muscle relaxation. So if you got that correct, good job. Our follow-up question 3 is, identify all 3 beta receptors and tell me where are they mainly located. So that's what I'm looking for, where are they mainly located. So the clinical utility of drugs which affect the adrenergic nervous system, well, you have the agonists of the beta 2 receptors, which are used in the treatment of asthma, right? Because it causes relaxation of the smooth muscles of the bronchi. That's A. For B, you have the antagonists of the beta 1 receptors are used in the treatment of hypertension and angina, because it slows the heart and reduce force of contraction. And then you have the third category where you have antagonists of the alpha 1 receptors, where they are known to cause lowering of blood pressure, right? So relaxation of smooth muscle and dilation of the blood vessels. So these are the clinical utility of drugs which affect the adrenergic nervous system. So I found this table that I just wanted to share, the alpha receptors versus beta receptors, right? So this is kind of a summary again, right? So here, alpha receptors are the cell receptors that control physiological processes like vasoconstriction, intestinal relaxation, pupil dilation upon interaction with epinephrine or norepinephrine. And the beta receptors are the group of receptors that control vasodilation, relaxation of the bronchial and uterine muscles, and increased heart rate. So the alpha, they stimulate effector cells, and the beta relax effector cells, right? clearly there's an exception to that. The two types are alpha 1 and alpha 2, three types, beta 1, 2, and 3, mainly occur in vascular smooth muscles and effector tissues, mainly occur in bronchi muscles, heart muscles, and uterine muscles. The alpha stimulates smooth muscles, the beta stimulate both cardiac and smooth muscles. So the methoxamine is an example of an alpha 1 agonist, while clonidine is an example of an alpha 2 agonist. Albuterol is an example of a beta 1 agonist, while metoprolol is an example of a beta 2 agonist. All right, so epinephrine or adrenaline is a hormone. It is a catecholamine, is sympathomimetic monoamine derived from amino acids phenylalanine and tyrosine. Epinephrine is sometimes shortened to epi in medical jargon. Epinephrine is now also used in EpiPens and Twinjex, etc. Right? So they are similar but contain 2 doses of epinephrine. The epinephrine can be injected directly into the heart to stimulate it after it has stopped beating due to drowning, suffocation, etc. The epinephrine dramatically restores the heartbeat. That's important. So in cases of shock, norepinephrine has been used to restore and maintain sufficient blood pressure and ensure adequate blood flow to vital organs. All right, great. So when local anesthetics are used to reduce or eliminate pain in a specific area, epinephrine is frequently used in conjunction with these agents to constrict the blood vessels. So just FYI, to have a general idea of the use of epinephrine in the real world, right? And we want to also look at the structure of epinephrine and norepinephrine that you should know. All right, so now we're looking at the life cycle of a neurotransmitter. So firstly, we have the synthesis of the transmitter, right? So we can say biosynthesis of the transmitter. Then secondly, you have packaging and storage in synaptic vesicles, right? So it is biosynthesized and then it's stored in synaptic vesicles here, right? If necessary, transport from the site of synthesis to the site of release from the nerve terminal. And then you have release in response to an action potential. So there's an action potential which triggers the release of the neurotransmitter. After that, step 5 says binding to postsynaptic receptor proteins. So we have the postsynaptic receptors here, the neurotransmitter binds. And then termination of action either by diffusion, destruction, or reuptake into cells. All right, so just a while ago we mentioned that our neurotransmitter must first be biosynthesized. So let's look at the biosynthesis of norepinephrine and epinephrine, right? So we're getting into some more chemistry here. So it starts off with L-tyrosine, right? So I do expect that you will know the biosynthesis of norepinephrine and epinephrine. Starts off with L-tyrosine. And in the presence of tyrosine hydroxylase, we form levodopa, right? And what is the difference between L-tyrosine and levodopa? Well, if you look closely, there's a hydroxide that's here. That's here. That is not here, right? So clearly, this hydroxylase catalyzes the hydroxylation of L-tyrosine to levodopa. So let's look at the next step, right? What can we observe as a difference between levodopa and dopamine? Well, let's look. So we see that our carboxylic acid here is missing, right? So clearly, dopa decarboxylase catalyze the decarboxylation of levodopa to form dopamine. So this is the structure of dopamine, which we have met previously. And then from dopamine to norepinephrine, what's the difference? What is the difference in structure? All right, well, if you guessed it, there's no hydroxyl group here, and there's a hydroxyl group here. So dopamine beta hydroxylase catalyze the hydroxylation of dopamine to form norepinephrine or noradrenaline. And then the final step in the biosynthesis of epinephrine now, since we have already biosynthesized norepinephrine, is the N-methyl transferase methylation, and it happens in the adrenal medulla, right? Notice the difference between norepinephrine and epinephrine is a methyl group, right? So this is a NH2, a primary amine, and this is a secondary amine with the methyl group here. So N-methyl transferase catalyze the methylation of norepinephrine to epinephrine. So that is how the body biosynthesizes norepinephrine and epinephrine. So this leads us to question 4, which says, list all enzymes involved in the biosynthesis of epinephrine. So if you can recall, write them down, or you can go back and write them down. All right, so the answer is tyrosine hydroxylase, dopa decarboxylase, dopamine beta hydroxylase, and N-methyl transferase. So our follow-up question 4 says, discuss the role of the four enzymes identified. So the 4 enzymes involved in the biosynthesis, let me know their role. What did they do? What reaction did they catalyze? All right, so we're going to go further into, the cycle of a neurotransmitter, all right? So we're just going to go a little bit deeper into that so that in the lectures which follow, we can understand, how these drugs interfere with our receptor, all right? We can have a deeper appreciation and understanding for how the drugs interact with our receptors. All right, so the first step is the storage and release, right? So as the slides progress, we will go little by little through this diagram, right? So a large percentage of norepinephrine present is located within highly specialized subcellular particles in sympathetic nerve endings and chromaffin cells. So much of the norepinephrine in CNS is also located within similar vesicles, right? So here we just went through the biosynthesis, right? This is the biosynthesis that we just went through, from tyrosine to L-dopa to dopamine to norepinephrine, etc., right? So once it is biosynthesized, it is now moved to the storage vesicle. All right, so the concentration in the vesicles is maintained also by the 12 helix membrane spanning proton antiporter called the vesicle monoamine transporter, which is VMAT for short. So following its biosynthesis and storage in the granules, the entrance of calcium, right, into these cells results in the extrusion of norepinephrine by excitosis of the granules, right? So depolarization and calcium influx. So anytime you see any sort of depolarization, usually that is related to ions going either entering or leaving. In this case, the ion is calcium, right? So let me read that again. Following the biosynthesis of the neurotransmitter and storage, the entrance of calcium into these cells results in the extrusion of norepinephrine by excitosis of the granules. All right, so carrying along with that, calcium triggered secretion involves interaction of highly conserved molecular scaffolding proteins, leading to docking of granules at the plasma membrane, and then the norepinephrine is released from the sympathetic nerve endings into the synaptic cleft. So this is the synaptic cleft, right? The space between the receptor and the neurons, right? So where are we? Where it's so norepinephrine is released from the sympathetic nerve endings into the synaptic cleft, where it interacts with specific presynaptic and postsynaptic adrenal receptors, right, on the effector cells. So it's released after the depolarization. It is released into the synaptic cleft, and then after that, it interacts with our adrenal receptors on the effector cells, triggering a biochemical cascade seen here that results in a physiologic response by the effector cell, which we saw, whether it's relaxation or contraction, that happens. That's generally speaking. All right, then you have uptake. So once the norepinephrine has exerted its effects on the adrenergic receptors, there must be a mechanism in place to remove it from the synapse and terminate its action at the receptors, right? Or it will continue to have that effect. So these are the three mechanisms that are involved in removing it once it has had its effect, right? So the first one is reuptake of the norepinephrine into the presynaptic neuron. In other words, it's recycling, which is a major mechanism. So after it has had its effect on the effector cell, right, there is recycling. So it now there's uptake, it goes back and eventually into some storage vesicle, right? So that is one major mechanism. It is there's the uptake or the reuptake of norepinephrine. The second thing that could happen is that you have conversion of the norepinephrine to an inactive metabolite, and in other words, this is metabolism. So we're going to look at that in a little bit. And diffusion, that's the third thing now, diffusion of the norepinephrine away from the synapse, right? So here we have diffusion and metabolism this way, right? So that's those are three ways or three mechanisms by which once the norepinephrine has its effect on the effector cell, it is removed in three ways, either reuptake or recycling, which is a major mechanism, or you have diffusion away from the synapse, or you have metabolism, right? So this arrow represents diffusion or metabolism. So now we're going to look at metabolism, right? Metabolism involves two key enzymes, and we need to know this metabolism. MAO and COMT, right? So norepinephrine, and we already know the structure of that. MAO is monoamine oxidase, right? It's an enzyme, right, that is involved in the metabolism of norepinephrine. So it converts the norepinephrine to an unstable imine, right? So if we look at the structure, everything is in place minus this portion, right? So after the unstable imine has been biosynthesized, then in the presence of water, we form an aldehyde, right? So the only change to the functional group has been occurring here, just here, which is why it's easy to know this metabolism, right? So we form the unstable imine here, and then after that, in the presence of water, you have the aldehyde being formed. And then the aldehyde is further oxidized to the carboxylic acid. And then in the presence of COMT, which is catechol O-methyl transferase, what do you have happening? We add a methyl. Well, let's look at it, right? The difference between this structure and this, the only difference is that we have the hydrogen has been replaced by methyl, right? So this is the metabolism of norepinephrine, and it involves two key enzymes, MAO and COMT, right? And eventually you just have, well, at the end of the day, you have a methyl here and the carboxylic group here. So both epinephrine and norepinephrine are orally inactive and have short durations of action because of their high hydrophilicity, ionization, and extensive first-pass metabolic deactivation by COMT and MAO. So COMT and MAO are very important for the metabolic deactivation of epinephrine and norepinephrine. The final carboxylic acid is polar and excreted in the urine. That is this one. All right, so in summary, we're summarizing now. Neurotransmission, it applies to adrenergic neurons innervating smooth or cardiac muscle, as well as synaptic connections within the CNS, right? So again, we have various diagrams saying the same thing, right? This is our CNS, autonomic, sympathetic, parasympathetic. We have our ganglionic regions, and we have our adrenal medulla. We have our neurotransmitters, adrenaline, etc. And we have our muscles here, which is different from our somatic division of our nervous system. All right, so continuing with our summary, noradrenaline is biosynthesized in a presynaptic neuron, then stored in a membrane-bound vesicle. All right, so you have biosynthesis first, and then it's stored in a membrane-bound vesicle, as we said, right? When a nerve impulse arrives at the terminus of a neuron, it stimulates the opening of calcium ion channels, as we said previously, and promotes the fusion of the vesicles with the cell membrane to release the noradrenaline, right? So here we have vesicles fuse and release transmitter, All right, so that is in response to the calcium calcium ion causing depolarization there. So the neurotransmitter then diffuses to adrenergic receptors on the target cell, where it binds and activates the receptor, leading to the signaling process, which will eventually result in a cellular response, right? So where are we? It binds to receptor binding a new signal, right? And then you have the neurotransmitter departs receptor. So after the message has been received, right, noradrenaline departs, right? So this is here. After the message has been received here, the neurotransmitter departs the receptor and is taken back into the presynaptic neuron by a transport protein, right? So that's one way in which we get rid of our noradrenaline, by the reuptake. Neurotransmitter reabsorbed by active transport. And then once in the cell, noradrenaline is repackaged into the vesicles. Some of the noradrenaline is metabolized before it is repackaged, but this is balanced out by the noradrenaline biosynthesis, right? So neurotransmitter repackaged or metabolized. All right, so this leads us to our final question, which is question 5. So it says, what are the 3 mechanisms for removing norepinephrine from the synapse? So once it has carried out its duties, what are the 3 ways in which it is removed? Pause. So if you answered, once norepinephrine has done its job, it's removed from the receptor by 1, reuptake of norepinephrine into the presynaptic neuron, or recycling, which is a major mechanism, by NET and into extraneuronal tissues. So NET means norepinephrine transporter, right? And then the second of the 3 is conversion of the norepinephrine to an inactive metabolite, or metabolism. And then the final way it is removed from the receptor is diffusion of norepinephrine away from the synapse. So as we said, the major mechanism is recycling, but there are 2 others. And then our final follow-up question 5 is, explain the statement, both epinephrine and norepinephrine are orally inactive and have short durations of action because of their high hydrophilicity, ionization, and extensive first-pass metabolic deactivation by COMT and MAO. All right, so think about it and come prepared to answer this question. So that's it for now. Thanks so much for watching to the end, and see you next time.